inheritance of two genes. Mendel also worked with and crossed pea plants that differed in two characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape, and one that had seeds of green color and wrinkled shape. Figure 4.7. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. Now here can you tell which of the characters is in the pairs yellow, green color and round wrinkled shape was dominant. Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those that he got then when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols. Y is for dominant yellow or seed color, and Y for recessive green seed color symbol, green seed color, red or capital R or round shaped seeds or small r for wrinkled seed shape. The genotype of the parents can be then be written as capital R, smaller capital R, capital I, capital I, and smaller, small y. The cross between the two plants can be written down. The capital R, capital I, and small y unite on fertilization produce the F1 hybrid, capital R, capital Y, small y. When Mendel himself hybridized the F1 plants, he found that third fourth of F2 plants had yellow seeds and one fourth had been green. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. 4.7 results of a dihybrid cross where the two parents differed in two pairs and contrasting traits, seed color and seed shape. Law of dependent assortment in the dihybrid cross, figure 7.4.7, the phenotypes are round yellow, yellow, round green, and wrinkled green has appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 it can be derived as a combination series of three yellow, one green, and three round and one wrinkled. This derivation can be written as follows: three round, one wrinkled, three yellow, one green, and nine is equal to nine round. Now yellow, three wrinkled, the yellow, three round, yellow is to one wrinkled green. Now, based upon now of such observations on dihybrid crosses between plants differing in two traits, Mendel proposed a second set of generalizations that we call Mendel's law of dependent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is dependent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of the two pairs of genes during meiosis and the production of eggs and pollen in the F1 capital R small y smaller capital Y small y plant. Consider the segregation of one pair of genes capital R smaller capital R and smaller. 50% of the gametes have the gene capital R and the other 50% have smaller. Now besides each important thing to remember here is that the segregation of 50% capital R and 50% of smaller is independent from the segregation of 50% has capital Y and the other 50% has small y. Similarly, 50% of capital R for capital R bearing gametes has a capital Y and the other 50% has a small y. Thus, there are four genotypes of gametes, four types of pollen, and four types of eggs. And the four types of capital Y, capital R smaller, and smaller capital Y, and smaller y, each with the frequency of 25% of one fourth of the total gametes produced. Now, when you write down the four types of eggs and pollen on the two sides of Punnett square, it is very easy to derive the composition of the zygotes that give rise to the two plants. Figure 4.7. Also, there are 16 squares. How many different types of genotypes and phenotypes are formed? Note them down in the format given. Can you using the Punnett square data workout the genotypic ratio at the F2 stage and fill in the formal format given. There is the genotypic ratio also 9 is to 3 is to 3 is to 1. Genotypic found in F2. There are expected phenotypes. Chromosomal theory of inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 19th. Firstly, communication was not easy as it is now. In those days, and his work could not be widely publicized. Secondly, his concept of genes or factors in Mendel's words, assistable and discrete units that controlled the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, through Mendel's work suggested that factors, genes, were discrete units. He could not provide any physical proof for the existence of factors or say what they were made of. In the 90s, three scientists Rice, Collins, and von Teischermark independently rediscovered Mendel's results on the inheritance of characters. Also, by this time, due to advancements in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes, colored bodies, as they were visualized by staining. Now, by 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Bowen noticed that the behavior of chromosomes was parallel to the behavior of genes and used chromosomes movement. Figure 4.8 to explain Mendel's law. Figure 4.3 recall that you have studied the behavior of chromosomes during mitosis, equational division, and during meiosis, reductional division. The important things to remember as that are that chromosomes as well as genes occur in pairs. The two alleles of gene pairs are located on homologous sites on homologous chromosomes. During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently of each other. Figure 4.9. To understand this, compare the chromosomes of four different colors in the left and right columns. In the left column, possibility one, orange or orange and green is segregating together, but in the right band column, possibility second, the orange chromosome is segregating with the red chromosomes. Saturn and Bovary ultra and that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors that carried certain and united. The knowledge of chromosomal segregation with Mendelian principles are called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis of variation. The sexual reproduction produced from origin worked with the tiny fruit flies, Drosophila melanogaster, figure 4.10, which were found were suitable, very suitable for each for such for such bodies. They could be grown on a simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single meeting could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscopes. Linkage and recombination. Morgan carried out several hybrid crosses in Drosophila to study genes that were sex linked. The crosses were similar to the hybrid process carried out by Mendel in peas. For example, Morgan hybridized yellow bodied, white-eyed females to brown-bodied, red-eyed males, and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio divided vary significantly from the 9 to 3 to 3 to 1 ratio expected when the two genes are independent. Morgan and his group knew that the genes were located on the X chromosome section 4.4 and saw quickly that when the two genes in a hybrid cross were studied were situated on the same chromosome, the proportion of parental gene recombinations were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on the chromosome, and the term recombination to association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. Figure 4.11. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, slowly slowed, very slow, very low recombination. Figure 4.11 across A. Now, while others were focusedly linked, showed higher recombination. Figure 4.11 cross B. For example, he found that the genes of white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2%. The recombination. His student Alfred Stratwent used the frequency of recombination between gene pairs of on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genomes, as was done in the case of human genome sequencing project described later. 4.11 linkage are results of two dihybrid crosses conducted by Morgan. Cross A shows crossing between gene Y and W. Cross B shows crossing between genes W and W hair dominant wild type, the eyelids are represented with plus sign as suppressive superscript. Note the strength of linkage between the W is higher than W and small m. Polygenic inheritance. Meta studies mainly described those unit limits, those traits that have distinct alternate forms such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have a tall or short people as two distinct alternatives, but a full range of possible heights. Such traits are generally controlled by three or more genes, and the and are those called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example of for his this in a polygenic trait, and the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. To understand this better, let us assume for that three genes ABC control skin color in human with dominant forms A, B, and C responsible for dark skin color, and are recessive forms A, B, C, A, B, and C for light skin color. The genotype with all the dominant alleles capital A, capital B, capital B, capital C, you will have a darkest skin color, and that with all the recessive alleles capital small a small b small c will have the lightest skin color. As expected, the genotype with the three dominant alleles and three recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in the genotype would determine the darkness of or lightness of the skin in an individual. Pleiotropic, we have so far seen the effect of a gene on a single phenotype or trait. Now there are there are however instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called a pleiotropic of gene. The underlying mechanism of pleiotropy is in most cases is the effect of gene or on metabolic pathways which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in the humans. So the disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydrolase single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and the reduction in hair and skin pigmentation. Sex determination. The mechanism of sex determination has always been a puzzle before the geneticist. The initial clue about the genetic chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetic chromosomal basis of sex determination. Hanking 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hinckley gave a name to this structure as the X body, but he could not explain its significance. Further investigations by other scientists led to led to the conclusion that the X-body of Hanking was in fact chromosomes, and that is why it was given the name X chromosome. It was also observed that in a large number of insects and mechanism of sex determination is of the XO type, that is at all eggs bear an additional X chromosome besides the other besides the other chromosomes, autosomes. Now, on the other hand, some of the sperms bear the X chromosome, whereas some do not. Eggs fertilized by sperm having an X chromosome become females, and those fertilized by sperms that do not have an X chromosome become males. Do you think the number of chromosomes in the male and female are equal due to the involvement of the X chromosome in the determination of sex? It was designated to be the sex chromosome, and the rest of the chromosomes were named as autosomes. Grasshopper is an example of type of sex determination in which the males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. And these observations led to the investigation of a number species to understand the mechanism of sex determination in a number of other insects and mammals, including man, XY the type of sex, and mammals, including man, XY type of sex determination is seen where both male and female have same number of chromosomes among the males, and X chromosome is present, but its counterpart is distinctly smaller than called the Y chromosome. Chromosomes both have males and females bear same number of autosomes, as hence the males have autosomes plus XY while the female have autosomes plus XX in human beings and in Drosophila. Now, the males have one X and Y chromosomes, now whereas females have a pair of X chromosomes besides autosomes. Figure 4.12ab. In the above description, you have studied about two types of sex determining mechanisms, that is XO type and XY type. But in both cases, males produce two different types of gametes, either with or without X chromosome, some gametes with X chromosome and some with Y chromosome. Such types of XX determination mechanism is designated to be the example of male heterogamity. In some other organisms, example birds, and a different mechanism of sex determination is observed. Figure 4.12c. In this case, the total number of chromosomes is same in both males and females, but two different types of gametes in terms of the sex. In summary, principles of inheritance and variation हमने पहले से देखा. जेनेटिक्स एंड डेवलपमेंट, इसमें हमने देखा मेंडल्स लॉ ऑफ इनहेरिटेंस ऑफ वन जीन. देन इसके बाद हमने देखा इनकम्प्लीट डोमिनेंस, को डोमिनेंस, इनहेरिटेंस ऑफ वर्ड टू जीन्स, लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट, linkages and recombination, polygenic inheritance, pleiotrope, sex determination. Then summary. Please like, share and subscribe. Thank you so much for watching.